Hi, good morning. My name is Charlene Salsiluneta, and my topic for today's video is Atasha telangiectasia. What is Atasha telangiectasia, or Lewy Bore syndrome, is an inherited autosomal recessive disease caused by a mutation of the Atasha telangiectasia mutated gene, or ATM gene for short. The canonical role of the ATM is to maintain the genome fidelity by facilitating repair following double-strand DNA breaks. ATM also plays a key role in redox control during oxidative stress. Loss of this two cellular function is believed to as the underlying source or cause of the AT or attached telangiectasia patient physiological and neurological symptoms. Patients with attached telangiectasia or AT lack the normal ATM control of oxidative stress due to reactive oxygen species, resulting in the degeneration of cerebral Purkinje cells. Neuronal cells are especially prone to oxidative stress damage in large part due to the higher levels of mitochondrial activity. Consequently, AAT patients undergo progressive neurodegeneration and suffer from effects of reduced cognitive function and loss of coordination and of fine motor control, as there is currently no established therapy for AT. A better understanding of the ATM dysfunction experienced by AT patients may shed new insight into the possible intervention or treating the cellular damage due to oxidative stress. This paper shows how protein to protein interaction may help to prevent or stop the two cellular function used by the by the ATM mutated gene. As it says here in the picture given by the Cleveland Clinic, by the AT symptoms include trouble walking, muscle twitching, slurred speech, and frequent infection. As said on the other slide, it is those the loss of its function may cause physiological and neurological damage. Introduction and background. Introduction. The approach of the researchers in this study to see how protein interaction when using the method of DSSO cleavable cross-linking technique to see how it could lead to prevent or stop AT. Firstly, it was used to evaluate the RMM-protein complex structure and the protein-to-protein interaction. Then, to elucidate the ATM binding partners, a cell undergo oxidative stress. Since the SSO cleavable crossing is a relative new approach, a purified mRNA complex sample was used to refine the technique initially. So it's still very new as of today. So we need fresh and very, very purified protein to make this work. To characterize the interaction ATM makes cells undergoing oxidative stress. Flag tag constructs of ATM were used to permit enrichment. Cells were then incubated with oxidizing agents, and while cells were still responding to the stress, the proteins were cross-linked. The cells were then harvested, lysed, and purified by the immunoprecipitation. The purified protein complexes were then prepared for mass spectrometry analysis. They were reduced, alkylated, and digested. The cross-linked peptides were then analyzed. The peptides were chronographically separated by a C18 reverse phase column analyzed using an orbital perfusion hybrid mass spectrometer, which contained the necessary firmware to perform the MSS cross-link instrument acquisition. As for the methods, the researchers used six main types of methods: protein expression and purification, ROS treatments of cells, health cell harvesting, protein cross-linking, XLMS acquisition and analysis, with using different type of spectrometers to scan the parameters, as said in the other slide. The proteins to see what the structure of the protein. For the tissue culture, cells were grown at 37 degrees Celsius, 5% CO2 in a humid incubator in the Bluesky modified H medium, supplemented with 10% fetal bovine serum and 1% strep. As for the ROS treatments of cells, the HEK or 293T cells were grown to 80% confluency on a 15 centimeter plate in 20 ml of media which was added to 10 microliter of 15 mm ASO3 solution for a final as concentration of 25 microm. and incubated over the night for 18 hours. The cells were then harvested. As for the results of the ATM Vivo cross-linking experiment revealed that while no relevant cross-linking were detected between ATM and other protein, specifically pathways were over and underrepresented by the COPM of ATM expressing cells constructs. Digestion of with trypsin yielded the most number of identifier crosslinks from the DDSSO cross-linking experiment. Of the 53 crosslinks detected in the experiment, 41 were found using trypsin as the digesting enzyme, and even revealing of the 115 detected peptides linked with the DSSO, 81 contained arginine residues at the C-terminus, while 3 had lysine G-terminus, indicating that the DSS cross-linker inhibited trypsin hydrolysis of peptide bonds adjacent to lysine. A small percentage, or 5% to be exact, of cross-linked peptides were cleaved at the N-terminus cross-linked lysine, which is suggested that a LYSN enzyme or peptidase metalloendopeptidase might give complementary results to trypsin. However, due to the high frequency of miscleavages, up to 5 miscleavages per individual peptide, the peptides were much larger and had multiple crosslinks or linker fragments and were unpenetrable by the X-link X algorithm. In fact, no crosslinks were successfully identified despite otherwise high percentage of crosslinks between the different proteins with the complex, and it's received the lowest X6 score of all time detected. The major drawback of this alternative enzyme was the much lower likelihood that the peptides contained their basic residues in their amino acid sequence and were thus not carrying the positive charge that they could be used to be detected by the mass spectrometer. As for the conclusion, protein complexes seem to be considered in the wild type of utilizing trifunctional cross-linking reagents. In this alternative approach of enhancement, protein complexes were formed with responsive gather covalently connected the cross-linker, such as biotin, rather than utilizing labels communicated on the cloned proteins. Additionally, different cross-linker lengths or other classes cross-linking reagent could be used to target different amino acid end residues. For instance, using cyplohydrin or carboxylic acid instead of amine-reactive NHS ester chemistry. And it says here that the protein, the DSSO, may not reveal secrets to the ATM, but it may lead to something that's still connected to the chromosomal breakage, just not ATM. But this is still a very new technology, a very new research, so there is still hope that it may be connected to a much lying deeper problem within the chromosomal breakage. And with that, thank you for listening to my presentation. Have a great day.